Welcome to the tiniest city you'll ever visit, the inside of a eukaryotic cell. Here, microscopic organelles are complex and are highly organized, each doing a unique job to keep life going. By the end of this tour, you'll know what every organelle does, how its internal parts work, and how they all work together. Things are about to get cellular.
Bill Nye the Science Guy. Bill.
First stop, the cell membrane, the border patrol of the cell. It's a phospholipid bilayer, two layers of molecules with heads that like water, hydrophilic, and tails that hate water, hydrophobic, which are lined up tail to tail. This structure makes it semi-permeable, letting some molecules through, like oxygen or carbon dioxide, while blocking others unless there's a special channel or pump. Embedded proteins act as gates, carriers, or receptors. Cholesterol molecules maintain fluidity, and carbohydrates serve as ID tags telling other cells, "Hey! I belong here." Inside lies the cytoplasm, a gel-like environment full of salts, nutrients, and enzymes. Organelles float here. It's the area where many chemical reactions occur, and the cytoskeleton threads through it like scaffolding, giving shape, internal structure, and routes for transport. The cytoskeleton is made of microfilaments, actin, intermediate filaments, and microtubules, which serve as cellular highways, tracks along which vesicles, organelles, and motor proteins travel. Next, the nucleus, the command center. It's enclosed by a double membrane called the nuclear envelope. This envelope has nuclear pores, specialized channels that regulate the flow of mRNA, proteins, and ribosomal subunits in and out. Inside, you'll find chromatin, DNA wound around protein cores, histones, floating in a fluid material called nucleoplasm. When the cell is about to divide, chromatin condenses into chromosomes. In the heart of the nucleus sits the nucleolus. This is where ribosomal RNA is assembled and combined with proteins to produce ribosomal subunits, which then exit the nucleus to form complete ribosomes in the cytoplasm. Because the nucleus holds the entire instruction manual, DNA, and orchestrates gene expression, it effectively controls all cell activities. Now let's go to the ribosomes, essential protein factories. Ribosomes consist of two subunits, large and small, made from rRNA and protein. They read the instructions from mRNA and link amino acids into polypeptide chains. You'll find ribosomes in two locations, free in the cytoplasm, producing proteins that function within the cell, or bound to the rough endoplasmic reticulum, producing proteins that will be secreted, inserted into the membrane, or sent to lysosomes. On the rough endoplasmic reticulum, these budding proteins enter the lumen, interior of the endoplasmic reticulum, where they fold and may undergo modifications such as addition of carbohydrate side chains. From there, they're packaged into transport vesicles that bud off and head to the Golgi apparatus. Adjacent to the rough ER is its smooth counterpart, the smooth ER. No ribosomes here. Instead, the smooth ER handles a variety of metabolic tasks. Lipid synthesis, making phospholipids, steroids, and cholesterol, vital for membranes and cell signaling. Detoxification, enzymes in smooth ER modify toxins and drugs to make them safer or more excretable. Calcium storage, in some cell types, like muscle, the smooth ER stores and releases calcium ions for signaling or contraction. Unlike the rough ER, the smooth ER doesn't send proteins outward, but communicates with internal membranes and supplies necessary lipids and detox functions. Proteins and lipids from the ER travel to the Golgi apparatus, the cell's postal service. The Golgi is a stack of flattened membrane sacs called cisternae. The process works like this. Vesicles from the ER arrive at the cis face of the Golgi. Inside the Golgi, cargo molecules are modified. Sugar side chains might be added, or phosphate groups attached. The Golgi sorts these molecules and packages them into new vesicles at the transface, then ships them to their destinations, the cell surface, lysosomes, or other organelles. The Golgi also generates certain vesicles that become lysosomes by packaging in digestive enzymes. Cells generate waste or suffer damage, so cleanup is essential. Enter lysosomes and peroxisomes. Lysosomes are membrane-bound vesicles containing hydrolytic enzymes. They digest foreign organelles, macromolecules, and even foreign invaders such as bacteria. They maintain an acidic interior to ensure enzymes function optimally and don't harm the rest of the cell if leaked. Peroxisomes specialize in oxidation reactions. They break down very long-chain fatty acids, detoxify harmful molecules like hydrogen peroxide, and convert it into water and oxygen using the enzyme catalase. Think of lysosomes as your cell's recycling center, and peroxisomes as its hazardous waste removal team. Now the powerhouse of the cell. Mitochondria. These organelles convert fuel molecules, like glucose and fatty acids, into usable energy, ATP, adenosine triphosphate, through a process called cellular respiration. Mitochondria have a double membrane. The outer membrane is porous to small molecules. The inner membrane is folded into cristae, increasing the surface area for metabolic reactions. Inside lies the matrix, which hosts enzymes for the Krebs cycle and other metabolic steps. Also present are mitochondrial DNA and ribosomes, meaning mitochondria can produce some of their own proteins. In sum, mitochondria are mini power stations inside each cell, turning food and oxygen into energy, carbon dioxide, and water. Now let's visit plant cells. They've got extra features that give them green. The cell wall. Outside the plasma membrane, plant cells have a rated cell wall made of cellulose fibers. This wall gives structural support, protection, and prevents overexpansion when water floods the cell. Tiny channels called plasmodesmata connect neighboring cells, allowing transport of nutrients and signals. Chloroplasts. These are the solar panels, organelles where photosynthesis happens. Chloroplasts have an outer and inner membrane. Inside, they contain sacs of thylakoids, forming grana, with chlorophyll pigment capturing sunlight. In the surrounding stroma, the light-independent reactions convert CO2 into sugars. Like mitochondria, chloroplasts have their own DNA and ribosomes, a hint at their bacterial origin. Central vacuole. Plant cells often have a huge central vacuole. It stores water, nutrients, waste, and gives the cell internal pressure to keep it rigid. When the plant lacks water, the vacuole shrinks, causing wilting. And that's the world inside the eukaryotic cell, a complex living system where every part works together in harmony. From the nucleus that holds the plants, to the mitochondria and chloroplasts that power it, to the membranes and cytoskeleton that hold everything in place. It's an incredible example of microscopic teamwork. So next time you say you're tired, just remember, your cells are still in there, working overtime,